shortest presentations or bios, introductions I've ever done, I guess. And let's see, where is Lucy and George at? So we need to change what we said. And I need, well, when I ask y'all, instead of doing it in the back of the room, come up the front of the room if you don't care, just FYI. And let's see,
Steve, do you want him to come down?
what now? Not yet, not yet. Let's see. Okay. So Steve Chyrchel gave me a real short one about his career, and it went along something basically 28 years interpreter for Arkansas State Parks, and life before that 30 years, and I wound up shaking the hand of Mother Teresa in the big house, I guess the White House, something like that. But anyhow, I'm like, no, I'm not doing that. You gave that responsibility to the wrong person. So here's the introduction I'm going to do for Steve Chyrchel. And so he has served as a park interpreter at Hobbs State Park Conservation Area for an impressive 28 years, playing a pivotal role in connecting people with nature, history, and conservation throughout Arkansas State Parks. Harry Truman once said, and I got this quote from Matthew Mobley. Where are you at, Matthew? You had said this before, and I copied from you on this one. And this is our own Missouri president right here. Harry Truman once said, "It is amazing what you can accomplish if you do not care about who gets the credit." And I tell you what, that embodies Steve Chyrchel right here. Steve embodies humility and leadership. He consistently delivers engaging, fun, meaningful nature and historical programs that create lasting memories for visitors of all ages. Beyond that, he uplifts and empowers those around him, often allowing others to shine while quietly being the driving force behind countless initiatives. One of Steve's most impactful contributions is the tireless work in restoring and raising awareness about the disappearing Ozark Chinquapin tree. He has become the voice for these once abundant native trees, educating thousands of park visitors about their cultural, ecological, and historical significance. Through his efforts, countless people have become inspired and mobilized to take part in his conservation success. Steve is not only a conservationist, but also a movement, a builder. He has inspired not just visitors, but an entire park system, and continues to champion Arkansas State Park's mission of stewardship and environmental education. Steve is a founding board member of the Ozark Chinquapin Foundation and remains actively involved with ongoing restoration plots and educational efforts. Steve Chyrchel, folks, is a true conservation legend of Arkansas and a powerful example of what quiet communication leadership can achieve for nature, for people, and for future generations. And we've got a little surprise for him too, and I can't say enough good stuff about it. So George and can y'all go ahead and bring this up the front? Got a little surprise for you, Steve. And Steve is real modest and real humble about everything. And this year, 2025, looks like it's going to be the last year that Steve works with Arkansas State Parks. He's retiring. We didn't want that to happen, but if y'all can unfold this, we got a little surprise for you. And happy retirement, Steve Chyrchel. 28 years of service. And he didn't want us to even say anything about this, but I'm not letting this go. We've got a table back there. We've got Sharpie markers, and anybody that wants to write something about Steve on this and we want to do it, you can fold that up and take that with you. And we just can't say enough good stuff about what you've done, buddy. Thank you. And we doctored that up. Yes, hats off to you. But anyhow, I'm going to turn it over to you now. And Steve Chyrchel has a really awesome program for us. Go ahead, buddy. Thank you.
I have a loud voice, so I'm going to stick to it. just look at some pictures. That's it. So I did that. Earlier this year, springtime, the University of Arkansas, it was a month long, did some very important research. And one of the things that happened while this research was going on was we had one of our volunteers come and take some pictures of some of these birds that were visiting the Ozark Chinquapin trees. And so, let's take a peek at those. I have to say, Ellen Proctor shot most of these pictures. This is an orchard oriole, and I wish I knew the answer to this, but I think I do know the answer. Of course, these are the male catkins, and they're not open yet, but they're still drawing insects, so I'm sure they're still emitting an odor which draws the insect, which of course draws the birds. Now, this next picture was taken by John Hughes. I love this picture. Got a nice picture. It's also an orchard oriole. And that orchard oriole had something on his mind. I'm going to go get that. That's a good picture too. Love that. Okay, then Ellen was looking at one of these orchard orioles. And that orchard oriole stared right back at her. And that orchard oriole came after her. And she got this picture. Pretty cool. All right, there were an awful lot of small birds. And if you picture this, here's Ellen, she's looking at these trees. So whatever's happening in the back, she can't see. Whatever's happening inside the tree, she can't see or photograph. So there were a lot of different small birds she was unable to get pictures of because of where they were and because they moved very fast. But there were some larger birds there. Of course, the northern cardinal. That wasn't me. Yeah. So the larger birds had a tendency to sit in one place, so Ellen got some good pictures. That's a white-eyed vireo. They move pretty fast too. Cute little dude, huh? Now, on the indigo buntings, the male indigo buntings, this was really interesting, stayed at the top of the trees and just sang and sang and sang, and I'm sure it was singing out for a girlfriend. So they spent a lot of time on the top of the trees. And I want to point something out here. Look at all these male catkins. This was loaded, so when they bloomed, they drew a lot of pollinators. And these male indigo bunnies just sat there at the top and sang, and sang, and sang. Now, Steve mentioned this earlier. Yes, sir. Oh, bye, Steve.
Gotta go.
Okay, Carol, bye. Steve mentioned this earlier, but I'm going to point this out again. Of course, we have our male catkins here, and they're starting to turn brown. That's not to say they're still not drawing a bunch of pollinators, because they are, and I'll go into that. But here, these little blooms, that's the female bloom, doesn't get very big. That turns into your coarser bird. I love pictures that tell you things. I love this picture. Because this was a small Ozark Chinquapin and it died. That told me something. Shoots are coming back. That told me something. It's a really nice picture. It was clear. And then, look at that. A bagworm. This plot was planted in 2014 and never has I seen a bagworm. And all of the other pictures that I'm going to show you, there are no more bagworms. And I come to find out, sparrows, wrens, finches, woodpeckers, warblers, and beetles eat them. So that's why we don't see them. They clean them up. Pretty nice. I went the wrong way, didn't I? This is a female indigo bunting. And what was interesting, this was taken in the tree, obviously, but Ellen said that the females spend most of their time on the ground. These are wire cages around our peon trees. And she said that the females spent their time down on the ground eating insects at that location. Of course, we had a lot of finches. In addition to things that they were eating in the tree, There's also thistle on the ground, which helped to draw them. Pretty birds, aren't they? So they were eating thistle, and every once in a while you get something extra, you get something special in a photograph. Isn't that cool? But what's really special is this. I love that. It was a clear shot of the bird, and boy, that finch was eyeing that bee. And then you know that they're there just to make new friends. Isn't that a cool picture? All right. Summer tanager. When we think of a summer tanager, we think of the male, we think of the red and the black. Sometimes we mistake that for a northern cardinal. But this is a female summer tanager. Female summer tanager. Female summer tanager. And talking about telling you things, I love this picture, because in the mouth of this female summer tanager, I'm going to guess, is a mealworm beetle, because there were a bunch of them on there. And the beetles that were there, they spent most of their time, all the different flavors of beetles, they spent their time mating. So they were laying eggs immediately, and the eggs would hatch, and you start going through your cycle where you have eggs, the larva, pupa, and adult. So what have you got to eat there? You got the beetles, and you've got the larva, which is the mealworm. So more stuff to eat. And I love this picture too. That female summer tanager is going through a gauntlet of insects, pollinators, to get to whatever she wanted to eat there. Look at all the insects that are drawn by these. Now these guys were kind of tough to photograph. This is a gray gnatcatcher, and they spent a lot of time on the inside of the tree. And this is why. They weren't going to the catkins. They were looking for beetles and larvae that was on the underside of the leaves. So we got a lot of activity, not just on the catkins, but all over the tree. Love the shadows there. Isn't that cool? This one picked off an inchworm, and I believe this was a mealworm, this pick. So even though these are not blooming now, there are larva stage of the beetles all over this tree growing those birds. These little guys move fast. This was a worm-eating warbler, yes. Only shot that Ellen could get of that. When they moved quickly. This was a yellow-breasted warbler. Right, can't miss those. And purple martins. Now, if you've ever been around purple martins, you know that they move fast. Ellen was not able to get a picture of these purple martins on the tree itself. The only shot she got, following them around, was one of them in the air. But purple martins also visit the Ozark Chinquapin trees. This was a yellow-billed cuckoo, and it stayed in the back. Ellen was up front, and I think this bird knew she was there. And so these yellow-billed cuckoos stayed in the back of the flock. Now, this was mentioned earlier, the northern parula, and there are a lot of them on these trees. But they move so fast, Ellen was only able to get 2 pictures, this one and this one. Hard to photograph because they're moving so fast. This guy, let's see, what was this guy? This is an eastern wood peewee. Trying to get these right, I want to tell you the wrong thing. Another one that was hard to, there's another shot of it. Cute, huh? Another one was hard to photograph, and this is not totally in focus because it was moving. It's a white-eyed vireo. This was very interesting. These are tree swallows. And she said there were 50 to 100, and they were, for a half an hour, they were going from one end of the plot to the other end of the plot eating. For a half an hour, they just kept doing it. Kind of interesting. This guy is what is he? He's an eastern phoebe. And then we have a hooded warbler. These guys are good looking, aren't they? Hooded warbler. The next one really turned out nicely. Isn't that nice? This next one here is a Kentucky warbler. Again, they wanted to spend their time in the middle of the tree, hard to get shots of them. Now this is a great crested flycatcher. Got to have your normal rim. You can talk to us a little bit there. And then there was, of course, this is a downy woodpecker because of all the insects. There were a lot of woodpeckers that showed up here. This is there's a female blue grosbeak. This one's a black and white warbler. And I want you to feature this. These are only 24 species here. There were a lot more that she couldn't photograph because they were too fast. But feature all that's going on. You've got all these pollinators on your catkins. And then these birds are there. And then in the back, way up there in that short-leaf pine, was this guy waiting to eat one of those birds. So, I mean, the whole story is there. Pretty cool. Well, you know, I can't just tell you, I can't just show you pictures of the birds. I've got to show you a few pictures of what they were eating. Of course, they were eating a lot of different stuff, and these are just a few. So, if you go to a picnic, and you have a picnic, what's the first guy to show up? Flies. They have an amazing sense of smell. Tim Johnson is a nature photographer with the Photographic Society in Northwest Arkansas. He shot these pictures of the insects. So these are different kinds of flies. I'm just going to go through these. Some were large, some were small, some were kind of weird. And then when you're at a picnic, you got all these flies. What shows up next? Ants. Ants have a terrific sense of smell. Not as good as flies, but they've got a great sense of smell, and that's why we see ants and flies on something to begin with. So, got some pictures of some ants. And of course, they're just other critters. Some of these guys look aerodynamically unsound, but they did all right. This is a European dark bee. That's just your bumblebee. Isn't that a cool picture? I like that. There's your honeybee. And the one I really like best, the honeybees, is this one. I really like that picture. Isn't that cool? I put that in there just for fun. It was just kind of a fun shot. But, you know, we think of bees as being excellent pollinators. There's why. They really get into their work. Really get into their work. And then the beetles, they were there just to mate. And of course, when they were mating, then the eggs would hatch, and there's larvae to eat. Boy, lots of stuff to eat here on these Ozark Chinquapins. I want you to remember, whoops, let's go back. Did I go back that far? From one side to the other, is only about a half an inch. So it's hard, it was very hard to photograph some of the pretty small guys in there. And of course your ladybugs are there. And every time you see a picture, you're going to see other critters there as well. Now remember I showed you that picture with that northern, not the northern, but there was a bird that was wanted to go underneath the leaf to get an insect. This is why. These critters are crawling around the underside of the leaf. The larva's there, they're eating that larva. And of course, they're all different kinds of critters. And these are just some. My goodness. And then the butterflies are not so much drawn by smell as they are by sight. Of course, the male catkins are nice and bright white and draws butterflies. So I got a few pictures here of butterfly. If you're wondering, is that a butterfly or a moth? Here's how you do it. You go to the antenna, these are like clubs, and they're different on each butterfly. If you have something that looks like a club, you got a butterfly. If you got something that's fuzzy like this, looks like an antenna, that's a moth. There were several different flavors of swallowtails. And here in this picture, again, I want to point out the female bloom. Not very big, they're very small. And although you have the male and female, of course you can't pollinate yourself. And then to end this up, Tim found a couple of butterflies that just wanted to pose. Isn't that cool? Weird looking guy. AJ, what'd you say that was?
A snout.
A snout, okay. That's a summer azure. And then I have just about 3 shots of this and I'm done here. But, I mean, this guy wanted to pose, and these are just cool shots. Oh, and don't forget your wasps. Now, you don't often think of wasps as pollinators, and they're inadvertent pollinators. Your wasps are there eating the larvae of the beetles. And so while they're in there, they're getting pollen on them, and they do some pollinating. So, I did my thing. I showed pictures, that's what Steve wanted me to do. But just showing you pictures doesn't tie it all together. You gotta have some meaning here. And Olivia Burdine is with us. I can give her a slide here. Look at that, Olivia, I'll give you a slide. Olivia Burdine is a PhD student at the University of Arkansas, Fayetteville. And she was the one who organized the research that went on this May and June at the Hobbs Ozark Chinquapin. So I want her to tell everybody how images like this fit into what she did this year, and then explain what you're going to be doing next year. So please give a hand for Olivia Burdine.
Hi, can everybody hear me okay?
Oh, you have to put your clicker.
It's there.
Forward, backwards. Okay, sir.
Push it hard.
Okay, so can everybody hear me now? Great. Okay, so speaking of pollinators, that's largely what I studied. So this can be a very quick presentation, just to kind of recap what I did this year and what I plan to continue to do to study the pollinators of the Ozark Chinquapin trees. So I'm characterizing the pollinators on the Ozark Chinquapin trees towards promoting their reproductive success. So first you may wonder, why would you need to study the pollinators in order to promote their reproductive success? Well, first we need to think about, what do pollinators do? We know that they facilitate pollination, but that's important because chinquapins cannot self-pollinate. So that facilitation of pollination enables the crossing of blight-resistant genes. And additionally, the more pollination that you have, the higher the seed yields that you have. Both the crossing of the blight-resistant genes and the higher seed yields are critical for the reforestation efforts. So, in the beginning, it was originally believed that Ozark Chinquapins were solely wind-pollinated. But in 2017, Chyrchel released a paper showing that insects were at least as important pollinators to chinquapin trees as wind was. And then, additionally, a PhD student named Olivia Burdine did a very short study on these same trees at Hobbs, characterizing the daytime pollinators. This was only about a week or so long. And so this is her data here. So if you see here, this is really important because you see most of our daytime pollinators, she started in the daytime, did not continue studying after dark. So she did not study nighttime pollinators. So if you notice here, we have a spike in activity and then the study ends. So that poses a big question. What's going on at night? So my main goal is to overall do a more in-depth study of all the pollinators, but really I wanted to shed light on the nighttime pollinators. So we know insects are important pollinators for the Ozark Chinquapin now. And we know that Lilia's data showed a big knowledge gap, and we don't really know what's going on at night. So this combined with the fact that the flowers of the Chinquapin trees are white, nighttime pollinators mostly forage by reflection and are highly attracted to white flowers, I have a pretty good idea that there's a lot more going on at night than anybody ever thought about. So also knowing that insects are at least as important as wind, but we've only studied the daytime. They may be even more important than anybody's thought about before. So just a quick little thing about Hobbs. I'm studying on 8 trees there. It's a planted plot of about 45, but 8 of them are seeding. And so my objectives are to determine the nocturnal pollinator diversity, as well as whether nighttime pollinators are different from the daytime, which they are very much. And also to figure out if any of the pollinators that visit the Chinquapin trees, daytime or nighttime, are different from the surrounding community pollinators. So general hypothesis is that pollinator communities will differ from nighttime versus daytime. Quickly about methods, I'm going to go over this very quickly because it can get really in-depth, and so if anybody has questions after this, come find me. So to collect the insects off of the tree so that I can identify them to species later, I used a few different methods. I used a kill jar and collected them by hand, putting the jar up on the catkins and letting everything fall in. And then I also used camera traps. So these are plant-insect interaction camera traps that are designed to video and collect images of plant-insect interactions. So I set these cameras up, built them by hand, programmed them, and set them up in the trees to capture video directly on the catkins. And so one thing that's important is to be able to have a physical sample to back up the video sample. So to do that, I actually use timed pet feeders. And so that's what this is. And so you see there's a little open cup here. I programmed these pet feeders to be able to be open for a certain window of time, 4 hours each. And so if I started the feeder at midnight, it would be open till 4 a.m. And then cup 2 would be open from 4 a.m. to 8 a.m. and so on. And so there was dish liquid and water in there. So that way I would know anything in cup 1 was from midnight to 4. So then when I go and look at my video and I look at midnight to 4 in the video, I know that I have a physical sample that matches that, and I know exactly who was pollinating when. And so too, I mentioned comparing the Chinquapin pollinators to the community pollinators. I did this by simply setting up some basic pollinator pan traps. Pollinators typically see white, yellow, and blue. I set up 3 of these 400 meters away from the main plot in different areas. And they ran 24 hours the whole time and collected them daily. So that way I'll be able to see insects that were pollinators but not attracted to the Chinquapins necessarily, and be able to compare those. That way we can be able to see if there's possibly a specialist pollinator. And so here's the fun part, we get to look at pictures of bugs. So these are all of our diurnal pollinators, and this is a very small selection out of however many I actually collected. There's many more species that I actually collected in the 1 month window that I sampled than what's pictured here. So just to kind of briefly, we have several different species of bees. We have bumblebees and sweat bees and mason bees and miner bees. In the second row we have syrphid flies or hoverflies. My mom always called them good news bees. We have thrips and fruit flies, blowflies and bee flies. In the last row we have click beetles, tumbling flower beetles, flower chafers, and brush-footed butterflies. And all of these are considered to be pollinators, and obligate pollinators, meaning they're there for the pollen. So these are a lot of our nocturnal pollinators, all different types of moths. On the top row, these are your leaf rollers and your geometrics, owlet moths, and ermine moths. And the bottom row we have plume moths. This is a very common, specific type of geometric moth. And then we have tiger moths and twirler moths. And so names like leaf roller or twirler usually refer to what they do to the leaves. It's often the larvae are twirling or rolling the leaves. And so these are insects that were also on the trees that are not pollinators. They do pollinate simply because they were found within the catkins, but they're not obligate pollinators. They're doing other things. So everybody up here, these three are predatorial hemipterans, and the one in the middle is a predatorial beetle. And then this one and these two are types of parasitoids wasps. So they're really good for things like biocontrol. It's why when you find pests on the trees, like these guys, they don't always become a problem. So when I was observing these trees, I saw lots of these, but the damage I saw was not significant, which again is thanks to critters like these. And then, so these are our herbivores and borers. And so, to conclude, my study again is going to identify pollinators of the Ozark Chinquapin tree across the full dial cycle, and I am hoping to identify any specialists of the Ozark Chinquapin tree, and then I plan to determine which species are the most prolific pollinators and when they are doing the most pollination. And then lastly, I want to provide data to conservationists that will allow for provisioning floral resources to the trees. Meaning that, if I know that the common eastern bumblebee, which I found a lot of, is really attracted to amaranths, then I will tell the conservationists at Hobbs, hey, these are really attracted to amaranths, and then they will plant amaranths to try and increase the amount of the common eastern bumblebee. And so going forward, I plan to expand my study. I want to find more sites and sample more trees. I do plan to continue observing the trees at Hobbs, but I would like more sites, some in a similar manner that are planted, and some that are more naturalized, so I can also compare the insect activity from naturalized plots to planted plots. And then I plan to investigate the treetops versus the tree bottoms, and I say that because one of the things I noticed was that the insects at the top of the trees were very different from the bottoms of the trees. And I'm not sure if that's due to insect foraging habits or if there's a difference in the trees. So that's also another problem and a bias in my current data. I was not able to collect out of the top of the trees because I'm not as tall as a tree. So I plan to come up with a way to sample the treetops as well. I will probably make some type of hanging track to do that. And then lastly, again, I want to create a catalog of the floral resources that should be provisioned to increase the pollinators. And so lastly, I just have a couple of acknowledgments. I first want to thank the Foundation for allowing me to speak today and share my research with you. And of course, Mr. Chyrchel for getting me involved with the Foundation and his continued support. And then, of course, the people in my lab who helped me collect data, as well as Arkansas State Parks and the Foundation for funding. So thank you. Do we have time for questions, or should we do that later?
No, we can go ahead. We're going to go on a break, I think, right, AJ, after this. Yeah, you can take questions.
Okay, does anybody have a question? Yes.
What would be the number one pollinator, in your opinion?
So the most abundant pollinator that I noticed?
Maybe the main one that does more of the pollination than others. What would be number one?
Okay, so I will say the most abundant that I saw were sweat bees and syrphid flies, and then at night the most abundant that I saw were likely your roller and twirler moths. This is again very preliminary, this study was just done, and I have not had a lot of time to go in and analyze and compare the samples to the time captured. So if I was going to make an educated guess, I would say it was going to be the ones that were most abundant, because those I saw not only just the most, like there were 20 of them in 20 minutes, I saw the syrphid flies and the sweat bees over a long period of time throughout the day as well. So I would likely say the hoverflies and sweat bees.
Thank you.
Yes.
I'm curious about something.
Yes.
When you were doing your nocturnal studies of the insects, did you notice many predators predating on the insects like bats and different things like that while you were out there?
I did notice bats at night. I couldn't see them, but I could hear them and they would fly over my head. And then also, other than that, I didn't see many nocturnal birds or anything like that. I did hear the bats quite often. And then there were also a lot of predatory insects still active at night, typically types of beetles like the orange and black cantharid, the soldier beetles.
I was wondering if you were able to, have you talked with anybody in the, I guess, the biology department, zoology or something? Would they be interested in combining with you to look at the mammals that are, the bats or something like that, to see species and all that? I mean, have you coordinated with, I'm just curious about that.
That would certainly be interesting. I have not talked with anybody yet. I know that there were some ornithologists doing some research, but I'm not exactly sure what their goals were, so I won't speak on theirs, but I know they were studying the birds across 24 hours as well.
Oh, wow, cool.
Yes.
Have you noticed any beetles actually eating the catkins, eating it itself, not pollinating it?
I did not notice, so you'll have things like the flower chafers that I mentioned, they're eating the pollen, but not necessarily the actual flower. But I did not notice anything eating the actual flower. So everything I saw was either eating the pollen off of the flower or was hunting other insects. Yes, sir.
And Olivia, what would you, so you work at night on the pollinators, obviously. And so whenever you would have rain, how did that influence the numbers of different, whole plethora of the different kinds of insects you were seeing? How would rainfall at night affect the activity you're recording?
Rainfall actually shortened my sampling by a lot, like at least a week. And the flowers are usually only out for a couple of weeks to a month, if you're doing, have a really good season. So the rain knocks pollen off of the flowers, and flowers don't always reproduce pollen if it gets knocked off. That could be it. So also, insects don't like rain. They're tiny, so imagine getting a gallon of water dumped on your head. You wouldn't want to be outside either. So unless it's just a very light sprinkle, insects often take shelter during rain. So there's not insect activity when it rains. And it rained heavily for like a solid week and a half during my collection, so that definitely put a damper on things. But again, I plan to continue studying next year. In addition to the insects taking shelter, some of the pollen would have been knocked off during those rains, shortening the sampling time as well as shortening the resources that the insects were after in the first place. Yes?
Is it still thought that wind pollination as well as insect pollination plays a large role?
The current consensus is that the most efficient pollination is combined wind and insects. But that's part of the reason I'm doing my study is, like I said, the current studies have only studied daytime pollinators and are saying, oh, they're just as important. So if we've only studied half of the 24-hour cycle and saying it's just as important, it's possible that they could be more important. But again, I'm not making a claim, I'm investigating that. So,
It's well established that wind pollination is effective and a
Yes.
significant part.
yes, that is the current consensus. Anybody else? Okay. Thank you very much for your time.